Okay, so we're gonna start with jaundice. Okay, so in a very fast manner. So metabolism of bilirubin and the liver is going to be done now. So let's get started. Pathophysiology of different types of jaundice. So the normal so we have two types of bilirubin, conjugated and unconjugated. This is a well-known fact. So like everybody knows that we have two types of bilirubin. One is conjugated, the other one is unconjugated, which is pretty clear now. Okay, so the property of a unconjugated bilirubin is that it is water insoluble. So it is insoluble in water. Unconjugated bilirubin is insoluble in water. So what happens is that the normal urine does not usually contain any bilirubin because the normal blood contains bilirubin in the unconjugated form. The unconjugated bilirubin is water insoluble because it is transported in the blood as a complex with albumin. So, like albumin is a plasma protein, the unconjugated bilirubin is just a carrier, just carried by albumin, and then the albumin bilirubin complex is formed. This albumin bilirubin complex is formed. It is not allowed to filter through the glomerulus. The unconjugated bilirubin is taken up by the systemic circulation by the hepatocytes. Unconjugated bilirubin is conjugated to bilirubin glucuronide by glucuronyl transferase within the liver cells, which are nothing but the hepatocytes. So this is basically the first step of conjugation. So conjugation is done to convert the bilirubin to bilirubin glucuronide, and this is basically the rate limiting step. The conjugated bilirubin is water soluble, and then what happens is that because it is not bound to albumin, it is filterable by the kidney. So like in case the urine is going to have some sort of bilirubin, it's going to be conjugated, especially because conjugated bilirubin is water soluble. And then the conjugated bilirubin, which is formed in the urine, sorry, which is formed in the liver. Now the conjugated bilirubin is formed in the liver, right? So it has to go to some other place. So it's going to go to the gallbladder in the form of bile. Well, so this bile is going to be excreted directly into the GIT, where the bile in the bile, it's going to be the bilirubin glucuronide is reduced to urobilinogen and stercobilinogen. Well, this reduction step is going to be done by colonic bacteria. Okay, which is why colonic bacteria is pretty important, one could say. Okay, so some of the bilirubin is just taken up by enterohepatic circulation. Well, when there is hepatocellular dysfunction or whenever the bile flow is completely obstructed, what happens is the conjugated bilirubin is going to leak out into the systemic circulation. So it doesn't directly reach the liver, but like it gets into the systemic circulation. So the conjugated bilirubin now can be excreted by the kidney and it is detected in the urine. So by and large, whenever we find a bilirubin in the urine, it's going to be a conjugated bilirubin, okay? Okay, so summary is that the conjugated bilirubin is the first one. It is the water insoluble one. And the bilirubin which is in the urine is water soluble conjugated bilirubin. Okay. Okay, so moving on to the bilirubin in urine, which is otherwise called bilirubinuria. Bilirubinuria can suggest of conjugated hyperbilirubinemia. Within the GIT, the conjugated bilirubin is degraded to stercobilinogen, which imparts the bad smell and the color for the stools. Urobilinogen, filterable by the kidney. Normally, a small portion of the stercobilinogen is reabsorbed from the GIT, excreted by the kidney as urobilinogen. So, non-obstructive hyperbilirubinemia, urobilinogen is present in the urine. For example, in case of hemolytic anemia, whenever like sickle cell anemia and all, or hepatocellular problems, then the urobilinogen is going to be present in the urine. Okay, and then moving on to two types of cells, Kupffer cells and stellate cells. Kupffer cells is nothing but a part of the reticuloendothelial system. It has phagocytic action and it lies in the sinusoidal vascular space. Part of the reticuloendothelial cell has phagocytic action. It lies in the sinusoidal vascular space. Okay. Moving on to the stellate cell. The stellate cell is nothing but a fat storing cell, otherwise called a ITO cell, ITO. And then it is located in the space of Dc. So this is overall about the pathophysiology of different types of jaundice. And we saw a lot of things about the bilirubin in the urine, which is nothing but bilirubinuria. And the other one is about the Kupffer cell and the stellate cell. The key feature is that the stellate cell is otherwise called the ITO cell or the fat storing cell. Kupffer cell is the phagocytic cell. And the rate limiting step is going to be conjugation of bilirubin to bilirubin glucuronide. Thank you.